glycolysis. In this glycolysis, if you see this diagram, here the glucose is converting to pyruvic acid. This glucose is 6-carbon compound. The pyruvic acid is 3-carbon compound. When the pyruvic acid in presence of oxygen, that will be entering into the aerobic respiration. And if it is absence of oxygen, it will be, you see that cytosol is nothing but the cytoplasm. So the all process is taking place here. The fermentation and the lactic acid, all these are taking place in the cytosol only. That means cytoplasm of the cell. The glycolysis is also taking place in the cytoplasm of the cell, but the Krebs cycle is taking place in the mitochondria. So the this diagram, we can clearly remember that. So the pyruvate is fork in the metabolic road. So it is the partial oxidation, breakdown of glucose to 2 molecules of pyruvic acid in presence of oxygen. It occurs in cytoplasm of all the living cells. The cytoplasm is the liquid substance that is present in the cell, that is also called as the cytosol. And these are the members of the scientists: Emden, Maynor, Pernas, and Mayoff. So here this is also a shortcut we can also call as the EMP pathway. In anaerobes, it is the only process in respiration. And now we are entering into the glycolysis. Here the glycolysis, the first substrate of the glycolysis is the glucose. Here it is 6-carbon compound. Here the 10 steps are there. The end product of the glycolysis is the pyruvic acid. So this glucose is converting into the glucose 6-phosphate by taking this ATP and giving rise to ADP. And this glucose 6-phosphate is converting to fructose 6-phosphate. And this fructose is again converted into fructose 1,6-bisphosphate. In the next slide, we can see that with the help of these enzymes, there's the 10 steps are there, 10 enzymes are there. And here there is the bifurcate of this fructose 1,6 into triphospho glycerol dehydrate 3-phosphate. This glycerol dehydrate 3-phosphate, these are the intermediate products that are performed seen in the C3 cycle. And triose phosphate, dihydroxyacetone phosphate. These are all the substance or the intermediate products in the C3 cycle. And again, they will be converted into this triose phosphate, again converted into glycerol dehydrate 3-phosphate. There is a mutual conversion will be taking place, and this is giving rise to triose bisphosphate and phosphoglyceric acid. This will be converted into phosphoglycerate and phosphoenol pyruvic acid. The end product is the pyruvic acid. This is the how the in every steps the enzymes will be utilized and the ATP is used and the water will be releasing, and the main is the end product is the pyruvic acid. So this is the mainly we can see in our books. So these are the different types of enzymes. These will be participating in the glycolysis. So the first the glucose is converted to glucose 6-phosphate, and this glucose 6-phosphate, fructose 6-phosphate by the enzymes like the phosphoglucose isomerase, and this fructose 6-phosphate is converting to fructose 1,6-phosphate in presence of phosphofructokinase. As a result, here when the kinase will be there, the ATP is taking and giving rise to ADP. And this fructose 1,6-phosphate bifurcate in presence of enzyme aldolase, it is giving rise to dihydroxyacetone phosphate and glycerol dehydrate 3-phosphate. This glycerol dehydrate 3-phosphate will be converting to different series, and last the pyruvic acid is formed. Again, this dihydroxyacetone phosphate converted to glycerol dehydrate, and again a number of series pyruvic acid will be forming takes place. That means from one glucose two molecules of pyruvic acid will be formed, and this total process is taking place in the cytoplasm of the cell. And here the two ATP molecules are formed here, and the two pyruvic acids and NADPH are also here. They are used. And here the three enzymes we have to remember this. These enzymes, they can be asked that the fructose 1,6-phosphate in presence of which enzyme it will be converted to dehydroxyestone phosphate and glycerol dehydrate 3-phosphate. That means aldolase. Or they may be asked that enolase. This type of bits they may be asking. And now the sequence of chemical reaction by which one molecule of glucose is converted to two molecules of pyruvic acid is called the glycolysis. This is taking place in the cytoplasm. It is called as the EMP pathway, and this is the common step for the aerobic respiration, and it includes the following steps. So in detailed study, if we learn that glucose react with ATP to form glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase, and this requires magnesium as a cofactor. So the glucose, by taking the ATP in presence of an enzyme hexokinase, is giving rise to glucose 6-phosphate and ATP. And now this fructose 6-phosphate is phosphorylated by taking this ATP to form fructose 1,6-biphosphate using an ATP molecule. It catalyzed by the enzyme known as phosphofructokinase and giving the ADP. This fructose 1,6-phosphate split into two molecules of 3-carbon compound, 3-phosphoglycerol dehydrate and dihydroxyestone phosphate. So they may be asked that glycerol dehydrate, that means that is a 3-carbon compound, and dihydroxyestone phosphate is a 3-carbon compound. This is catalyzed by the enzyme aldolase. Again, fructose 1,6-phosphate converting to 3-phosphoglycerol dehydrate, and these two compounds, glycerol dehydrate and dihydroxyestone, that means that two compounds are formed here by an enzyme known as aldolase. That means bifurcate of fructose 1,6-diphosphate is taking place. That we have to remember the enzyme as aldolase. And these two compounds are interconvertible. So what we have to remember is they are interconvertible. They may be converted to 3-phosphoglycerol dehydrate to dihydroxyestone acetone, or this dihydroxyestone is converted to phosphoglycerol dehydrates. And 3-phosphoglycerol dehydrates give rise to dihydroxyestone phosphate. And now 3-phosphoglycerol dehydrate react with phosphate, NAD phosphate, in the presence of NAD to form 1,3-diphosphoglyceric acid and NADH2. This reaction is catalyzed by the enzyme phosphoglycerol dehydrate dehydrogenase. That means NADH2 is coming outside. So it is dehydrogenase. And the 1,3-diphosphoglyceric acid react with ADP and loses a phosphate group, forming 3-phosphoglyceric acid. This reaction is catalyzed by the enzyme phosphoglyceric acid kinase, magnesium acting as a cofactor. They may ask that this phosphoglyceric acid kinase enzyme is consisting a cofactor like magnesium. One ATP is formed from ADP. So, and this phosphoglyceric acid is converted to 2-phosphoglyceric acid in presence of enzyme phosphoglucomutase as a magnesium as a cofactor. And now the conversion of phosphoenol pyruvic acid to pyruvic acid is the final step of the glycolysis. So the end product of the glycolysis is the pyruvic acid. This reaction is catalyzed by the enzyme phosphopyruvate kinase. The magnesium and the potassium are the cofactors for the reaction. They may be asked this magnesium, these inorganic substances which are utilized as the cofactors in the enzymes. So the pyruvic acid thus enters the citric acid cycle. And the pyruvic acid is the 3 carbon compounds that will be entering into the citric acid cycle or the that is Krebs cycle. And significance of glycolysis. In glycolysis, 4 ATP molecules are formed by phosphorylation and 4 ATP molecules are formed by terminal oxidation. In glycolysis, from 1 molecule of glucose, 2 molecules of pyruvic acid, 6 molecules of ATP are formed here. 2 ATP molecules consumed in the reaction for the glycolysis. So the 2 NADH formed during glycolysis in 4 ATP molecules by oxidative phosphorylation electron transport system. So thus in glycolysis, 6 ATP molecules are net gained. Glycolysis is the common to both aerobic and anaerobic kind of respiration. So here, after this glycolysis, they may be entering into the Krebs cycle or the citric acid cycle. This pyruvic acid is converting into acetyl coenzyme A and entering into the mitochondrial matrix for this Krebs cycle. Okay, sir. Thank you. Any doubts you can ask me, students.
Okay, thank you very much, madam. Really wonderful presentation. Plant physiology, one of the most important topic. Madam has clearly, elaborately explained the total cycle or total pathway, EMP pathway is clearly explained. So what type of enzymes are utilized and which type of enzymes are involved, which reaction is most important, ATP synthesis and ATP utilization of the ATPs. So glycolysis is one of the most important topic. And also, madam has clearly explained about the structure of the mitochondria and the difference between aerobic and anaerobic respiration. Clearly explained about the formulas of the aerobic and anaerobic respiration. So most of the topics related to this EMP pathway, most of the questions are definitely you will look into. You're going to attend the examination. RQ, respiration quotient, is also most important. So oxalic acid RQ value is 4. Remain a carbohydrate 1 and lipids and fats 0.7, protein 0.9. So these are the RQ values also most important. At the same RQ formula is also most important. And even though Madam has clearly explained why we can also call the EMP pathway and what are the steps involved, the balance sheet of the ATPs, synthesis of the ATPs and ATP consumed in glycolysis or EMP pathway. Madam has clearly explained, my dear friends. So this is the most important topic, not only in CPGET, but also all competitive exams related to plant sciences. So I hope you are enhanced, your knowledge has enhanced through our respected madam class. Really wonderful, madam. Really, I am also listen each and every point clearly explained. Thank you, madam. Prakash, any doubts?
Sir, can you send me that after completion of entrance exam, botany question paper? Entrance exam, botany question paper.
Definitely. After previous papers.
This on 8th we have botany exam.
9th we have PG entrance. Definitely will send you. Don't worry.
So that's why I calling, sir. You
Sure,
are not
sure, my dear Prakash. Will send you.
Sir, group lo send cheyandi, sir.
Thank you.
Yeah. In our group, definitely will send you.
Thank you so much, sir.
Right. First of all, you should thank our respected madam.
Madam, thank you so much.
Thank you, sir. Thank you.
Right.
Sir, exam ఎప్పుడు సార్ పిల్లలకి?
Exam 9th రోజు ఉంది మేడం. 9th రోజు ఉంది. నేను అంత లోపల వీళ్ళకి మ్యాక్ టెస్ట్ లోనే ఒక మాక్ టెస్ట్ లో ఒక 3 తయారు చేశాను. అంత లోపల ఈ ఆన్లైన్ ఈ ప్రాక్టీస్ టెస్ట్లు తర్వాత అసైన్మెంట్లు పంపిస్తున్నాను. ఇవి కూడా యూజ్ అవుతాయి. తర్వాత వీడియోస్ కూడా ప్రీవియస్ నేను అనాలిసిస్ చేసిన పీజీ ఎంట్రన్స్ వీడియోస్ కూడా పంపించాను. అవి ఇప్పటి వరకు ఒక 8-9 పంపించాను. ఇంకా 10 ఉన్నాయి. అవి ఇవాళ 2, రేపు 2, ఎల్లుండి 2 పంపిస్తా. ఒకటేసారి మొత్తం అంటే వాళ్ళు కన్ఫ్యూజ్ అవుతున్నారు. మేడం థ్యాంక్ యూ. టుమారో
Thank you.
సేమ్ టైం విల్ గోయింగ్ టు మీట్.
Okay, thank
థ్యాంక్
you.
యూ మేడం. థ్యాంక్ యూ.
Thank you very much.